Ribonucleic acid, or RNA for short, is a name given to the thesis chain-based sequence required in many activities within the cell. RNA, consisting of 5 carbon ribose structures, is also extremely important in terms of DNA. What is RNA, what does it do, and where is it found? We have compiled detailed information about the properties and task of ribonucleic acid. Understanding RNA structure primarily requires the study of ribose and DNA structures. Different types of RNA found in the cell have different tasks. What is RNA? In order to understand RNA, ribose and nucleic acid structures must first be understood. The smallest building block of carbohydrates in the body is called monosaccharide. Monosaccharides cannot decompose into smaller building units. Ribose is also a type of monosaccharide and therefore cannot be decomposed into smaller pieces. Ribose in the pentose group consists of a combination of 5 carbon atoms. Nucleic acid, on the other hand, is a structure consisting of nucleotides that are found even in all living units and viruses. RNA contains a nucleotide consisting of a phosphate base, a nitrogen base, and a ribose. This nucleotide is called ribonucleotide. RNA is a biologically extremely important molecule and occurs by chain binding of nucleic acid consisting of a nitrogenous base, phosphate base, and ribose group. What does RNA do? Where is it located? Different varieties of RNA are found, and different types of RNAs have different tasks. 1. Ribosomal RNA, RNA. Ribosomal RNA is located in the ribosome part of the cell. 80% of the RNA present in a cell is RNA. Ribosomal RNA is used to form ribosomes that are filled with enzymes used for protein synthesis, and their synthesis is carried out in the nucleus. For this reason, RNAs, which combine with proteins in the cytoplasm, also play a large role in the formation of amino acids. They can bind to other molecules and turn as 2. Messenger RNA, mRNA. Messenger RNA accounts for 5% of the total RNA in the cell. It plays a large role in copying the genetic code in DNA and transmitting the message received from DNA. mRNA is the first discovered RNA variant. It is a structure that allows it to be copied by taking the information necessary for the type of protein to be produced from DNA. A gene that carries information about the protein to be synthesized allows mRNA to be synthesized. 3. Carrier RNA, tRNA. The smallest building unit among RNAs, is a structure transferred by amino acids. It acts in the transport of amino acids necessary for protein synthesis from the cytoplasm to the ribosome. Consisting of approximately 80 nucleotides, tRNA becomes a 3D structure by combining hydrogen bonds with bases. During protein synthesis, tRNA is a structure that can be used repeatedly and repeatedly. It is done again by breaking down when its structure breaks down.